On behalf of Nuvisan, I would like to welcome you to our talk today. I'm going to talk about how a sophisticated study design of a regulatory subinduction study may help you in terms of DDI risk assessment. But before we explore our today's topic in more detail, I would like to introduce myself very briefly. There is an issue with the, oh no, it works. My name is Kathleen Römer, and I'm working as a project manager in the PK science team of Nuvisan. Our group was founded to consult clients in various DMPK aspects throughout all phases of the R&D value chain. My personal work experience started 15 years ago in the field of toxicology, but already a few years later, my work focus turned into the direction of DMPK, and it is still a passion for me to work in this field. Throughout my career, I could gain a profound knowledge in diverse techniques such as gene expression profiling, but also cell-based in vitro screening approaches for the tox and PK characterization of drugs and chemicals. Before we start, some important notes up front, so-called house rules. Your microphone is muted by default. If you have a question, please use our Q&A box. So not the chat box, please use the Q&A box, type in your inquiry, and after the talk, I will endeavor to answer as many questions as possible. Please also note, this session will be recorded and is available afterwards on our website, and you can find it in the events category. Who are we? Nuvisan is a fully integrated contract research and manufacturing organization. We are a bit more than 1,000 employees distributed over 6 locations, and we offer services along the entire drug development chain, starting from target identification till early clinical trials. The regulatory subinduction study is part of in vitro PK characterization of a drug candidate and usually performed during non-clinical development, indicated here by light blue arrow. But we can not only support regulatory studies, we have also a subinduction screening assay and an innovative approach, a so-called PXR co-crystallization technology, which can be used to design out PXR and hence 3A4 induction activity very early during drug discovery. Here you can see our today's agenda. You have already heard about our services, and I would like to introduce subinduction as such a bit in more detail, some mechanisms behind, and provide you a clinical relevant example for a drug-drug interaction based on an induction mechanism. In the next part of the talk, I would like to show you our Nuvisan standard assay design and explain the principle of the branched DNA plex technology for mRNA quantification in more detail. Afterwards, I would like to show you some experimental options you may choose depending on your drug characteristics. And finally, I would like to point out how these experimental options may help you in terms of DDI risk assessment. And then we have time for a Q&A session. Subinduction, what does that mean? The primary organ of biotransformation is the liver, and quantitatively, the most important family of drug metabolizing enzymes are the cytochrome P450s. But the cytochrome P450s can not only metabolize drugs, it can also happen that drugs alter the expression and activity of drug metabolizing enzymes. And how subinduction works on a cellular level is shown on this slide here. A perpetrator drug can bind to certain cellular proteins, here from the nuclear receptor family, which are ligand-activated transcription factors. After dimerization, they translocate into the nucleus and can bind there to specific elements in the DNA and hence drive the expression of the target genes. And among these are our drug metabolizing enzymes. In terms of subinduction, the most relevant isoforms are SUB1A, CYP2B, and CYP3A. They are controlled by different ligand-activated transcription factors, the so-called aryl hydrocarbon receptor, constitutive androstenedione receptor, and the Bragnin X receptor. Please note there is a significant crosstalk, in particular between CAR and PXR, which means they have an overlapping spectrum of target genes for which they control the expression. The consequences of SUB-induction can now be manifold. This means if you increase the expression and hence the activity of a cytochrome P450, this may lead either to a decreased systemic exposure of the inducing drug. If the inducing drug is also metabolized by the enzyme which it induces, this is a so-called autoinductive mechanism. The second scenario which can happen is that you observe a decreased systemic exposure of a co-administered drug, if the co-administered drug is metabolized by the enzyme which is induced. And the third scenario could be that you observed an increased exposure of active or toxic metabolites. A clinical example is shown here, and I have chosen a combination of rifampicin and midazolam. Our victim drug here, midazolam, is a medication used for anesthesia and procedural sedation, and it is extensively metabolized by CYP3A4 during its first pass. Our perpetrator drug, rifampicin, is an antibiotic, and it is the most powerful known inducer, and it has a strong effect on CYP3A4 in particular. And by means of increasing the expression and activity of CYP3A4, you increase the metabolism of midazolam and hence reduce the concentration of the pharmacologically active parent drug. And you can see in the plasma concentration time profile indicated here by the filled dots that in the co-treatment situation, the area under the curve is reduced by about 96% compared to the treatment with midazolam alone. This means oral midazolam is completely inactive when co-administered with rifampicin, and this holds true even if you increase the dose. For this example, you have to think about alternative therapies. It's for sure an extreme example, but it shows how pronounced such an interaction can be. How can you now measure SUB-induction? You would usually start in drug development to measure SUB-induction with an in vitro assay, and the gold standard cellular model are primary human hepatocytes. We use 3 single donors, and we pre-characterize our donors regarding their inductive responses. We culture our cells in a so-called sandwich format, which means that the cells are embedded into 2 layers of collagen. We start the treatment by addition of the test compound of interest, usually at 7 concentrations and 3 replicate wells. We also include positive and negative controls in our design. Positive controls to show that the test system was inducible on the day of the experiment, and negative controls to show that the effect we observe is really test item specific and not just occurs by means of the treatment. Positive and negative controls are run at 1 concentration only, but again, 3 replicate wells. The entire treatment period lasts 48 hours, and we replace the treatment medium with the compound daily. On the right side here, you can see our analysis endpoints. Throughout the entire treatment period, we monitor morphology and confluency. And in addition, at the end of the treatment period, we perform a cell viability assay. Our standard assay is the cell title blue assay. And we perform a cell viability assay because we would like to identify false negatives, which means wells which show no induction, but only due to a cytotoxic effect. Our second analysis endpoint is the measurement of the test item concentration in dosing solutions and spent media. Spent media is usually measured during last day of treatment at 4 different time points, and all these measurements are performed routinely by UPLC MS-MS. We perform the concentration measurements because we have to show that our test system was exposed throughout the treatment period. In addition, we measure stability in all solutions because we have to ensure that everything we measure is measured within a predetermined stability timeframe. And our actual endpoint is the mRNA quantification. And we quantify the mRNA of CYP1A2, 2B6, and 3A4, along with 3 housekeeping genes by the quantiGeneplex assay technology. A typical result is shown here. We plot fold induction over solvent control against the 7 test item concentrations. And you can see here that our example test drug is a significant inducer of CYP3A4 in this particular signal donor at the high concentrations. On the right side, you can see some selected historical data from our test facility for 1 single donor. And we use as positive controls for the assay omeprazole for 1A2, phenobarbital for 2B6, and rifampicin for 3A4, and flumazenil serves as negative control for all CYPs. An important note here, why do we measure primarily mRNA quantification and not enzyme activity? The reason is that it can happen, and it is not so rare, that the test item is not only an inducer, but also an inhibitor of CYPs. And in such a case, the induction may be masked on activity level, and also the authorities recommend to measure mRNA as a primary endpoint. I have mentioned that we use the quantiGeneplex assay technology. This technology is not brand new in the meantime. It has its roots 20 years ago, and it is now well established, and numerous publications are out there showing that it's highly comparable to the gold standard qPCR or also array-like technologies. And the assay works by addition of a lysis buffer to your cells at the end of the treatment period to release the RNA. You can then use your lysates directly further. There is no need for RNA purification or amplification. You just add the lysate to a new well together with a customized kit. And the customized kit contains of a customized oligonucleotide probe set and Luminex beads. These beads are fluorescent microspheres, and they can be distinguished by means of their diameter and differences in their fluorescence emission spectra. And you can design your probe set in a way, and your beads in a way that, for example, the dark red bead binds only 1A2 mRNA, and the orange one binds only 3A4 mRNA. And after you have then performed the capture step, you will do a signal amplification by fusing a DNA amplification molecule to the mRNA, which is bound to a certain bead type. And this is the reason for the name of the technology, branch DNA technology, because this DNA molecule here looks like a tree, and it has several branches, and each branch has multiple binding sites for a fluorescent label. And this gives you a massive signal amplification and allows you to work with a very low amount of starting material. The last step is the readout, and this is performed with a Luminex device, and independent which device you use, the basic principle is very similar. You have 2 lasers. One will identify the bead type, and the other will identify the fluorescence intensity associated to a certain bead type. And the median